Continuing chapter 7. Those are supposed to be sperm heading toward an egg cell. One of them has a real old-fashioned outboard motor attached to it, saying, see you around, guys, and laughing. We've talked about the differences between male and female. We talked about how sex cells only have one set of chromosomes in them. Now we're going to talk about how you make cells, or nuclei, with just one set of chromosomes. The process is called meiosis. Now, the way I learned it, because I have a terrible memory, remember, and I wanted to be able to remember one from the other, is that mitosis produces two cells. It's the one with the T in it. Meiosis produces sex cells. It's the one with the E for sex in it. Whatever works. But in both cases, the setup is, let's make a copy of all the DNA. So there's two sets of chromosomes always in eukaryotic cells. So now you have made 4 sets. Well, for mitosis, you're going to get 2 sets and 2 sets to duplicate the original cell. In meiosis, to get just 1 set of chromosomes, you need to make 4 cells, each of which, or 4 nuclei, each of which is going to get 1 set of chromosomes. So that is a major difference between meiosis and mitosis. And it kind of leads to a lot of the other differences as well. This is meiosis. Production of gametes is the typical, the actual term, but we're going to call them sex cells. I do try to avoid terminology as much as I can get away with. And DNA replication. If you're going to start with 1 cell and wind up with 4, I suppose in theory you could set up 4 nuclei and divide the cell 4 ways. And I'm sure somewhere there's things that go through meiosis that do that. But for most living things that do this, they go through 2 cell divisions. They go through Meiosis I and Meiosis II. And I will write some of this down, but I'm going to show it to you. Meiosis I is always written Meiosis I, and Meiosis II is written Meiosis II. Well, not always. Sometimes it's the number. But it's very typically I and II, but it's always called 1 and 2. What you're going to do that's different is your first division is going to get the sets away from one another, but they're going to leave the copies alone. So we've got 46 chromosomes. They're all copied, but they're still attached to one another. So in theory, we still have 46 chromosomes, but we have 2 sets of 23. For meiosis I, we're going to put all the matching chromosomes up with one another, so that we can set them up and get them apart. And then once we've got them apart, we make 2 cells, then we can get the copies away from the copies. It is that simple. So the second part, the second division, looks like mitosis, except with only half as many chromosomes. So our chromosomes are numbered 1 through 22 in X and Y. So at some point during meiosis I, the two number ones are next to one another, copies, each one's copies, but the two number twos, the two number fives, the two number 15s. If you're female, the two X's are put next to one another. If you're male, the X and a Y are put next to one another. Even though they're not matching, they kind of pair up anyway. And it takes a while. Not entirely sure how they match them, but they can hang out in the middle for a while, waiting for all the other ones to get in line. And they're very close to one another, and they can, and their molecules are all moving, and they can touch each other. And a weird thing happens when they touch each other. They can trade pieces. They can trade ends. So imagine you're in a crowded elevator, and you happen to bump the person next to you, and when you step away, you've given them the bottom of your leg, and you've got the bottom of their leg from the contact point. You've just traded pieces. That is called crossing over. Remember when the guys followed up on Mendel, and they found out not all the chromosomes, not all the genes are separate from one another. Some of them are attached to each other, and they came up with the term of linkage. Now, they didn't know that they were molecules on chromosomes. They just knew they were somehow connected to each other. And so they did a whole bunch of research, and they did a bunch of crosses, and they found out, doggone it, they don't always stay linked. Sometimes the linkage breaks, and they swap over. Well, what it turns out happens is the two linked genes on one chromosome, one of them winds up on the sister, and the other one's over here. So say this was red hair and green eyes, and this was blonde hair and blue eyes, is you could swap, and the red hair would be with the blue eyes, and the blonde hair would be with the green eyes. And if you take a genetics class, how often that swap happens has a lot to do with how far apart the genes are. There's more places to swap the farther away they are. And you can actually count how many times crossing over happens and decide whether two genes are really close together or really far apart. Math. But there's only really one other thing about crossing over that I want to mention, is this also does something that was a mystery for a very, very long time in when people started to apply genetics to evolution, which is generally as you come down a family tree, there seems to be more genes and more DNA. It's like, where does it come from? And then an old question was, okay, so evolution happens when you get mutations in genes, and the genes take on a new use. To which you may go, but didn't you kind of need the old use? How do you get if they change, don't you lose what they used to do? And that seems like that would make it much harder to develop any kind of new use. Here's the deal. Imagine you're standing next on the elevator to somebody who is much taller than you, and you touch legs, but when you step out, you've only got their ankle and foot attached to your leg. They've got your entire leg from the hip down attached to theirs, attached to their ankle. So they have your knee and their knee still. They have your shin and their shin still. They have the bottom of your thigh and the bottom of their own thigh. They have part of their leg and a whole bunch of your legs the same pieces. Crossing over is not always equal. So you can cross over and give the other chromosome extra copies of a gene it only had one of. So it can keep using one gene and use what it was doing previously, but if the second copy mutates and that protein takes on a different purpose, that's great if it's useful, but you haven't lost the old purpose. And that's why if you look at the genes for spiders, look at a chromosome. The gene for digestive enzymes and toxin, what they inject when they bite into, they poison their prey. The genes are very similar to one another in terms of DNA sequence, and they're pretty close to one another on the same chromosome. At some point, there were digestive enzymes, and there was an unequal crossing over, and one of the chromosomes wound up with a digestive enzyme attached to the chromosome where they already had a digestive enzyme. So they kept using this one, and this one mutated into something a little bit less digestive and a little bit more neurotoxin. Pit vipers, do you see the same thing? The linkage, you not only see evidence of this unequal crossing over because the genes are really close to one another, but doing different things. But if you compare the sequences, they are homologous. They have very similar sequences. It's just changed enough that it's a protein doing a different thing. And that was a huge mystery kind of solved. Here, they're using different colors to represent one side and the other. And they cross over, and you wind up with part of the blue chromosome on the red one and part of the red one on the blue one. And then you wind up with a sex cell with a mixture. You're not going to get them in all of the sex cells because it hasn't crossed over with both copies. But that's all you need for that to be the one that forms the next generation. And now you've got unlinkage where you used to have linkage, and sometimes extra genes where you used to have genes. So in, sorry, I didn't mute this. So in Meiosis I, the sets get pulled together next to each other, hang out there, cross over. And during anaphase, the copies don't separate, the sets do. So in humans, we've got 23 matched with 23, duplicates attached to duplicates, and the sets go away, but each of the chromosomes is still a duplicate attached to a duplicate. So in Meiosis I, that's the big difference. The division separates sets, and then you go from there. In Meiosis II, if you watched a human cell doing that, it would look like mitosis. The difference being, instead of 46 chromosomes, there'd only be 23 where the duplicates, the copies separate from the copies. So you wind up with two cells, each with a set of duplicate chromosomes, and then each of those form two cells with what they call single-strand chromosomes. And now you got 4 cells. Now, if you're making sex cells, they're all going to be usable sperm. If you're making sperm cells, all going to be usable sperm. If you're making egg cells, the division holds all the food in one of the cells and then just separates. You got to get rid of the other chromosomes. The other 3, that's where the other 3 polar bodies come from. Little bitty cells purely to get the extra chromosomes into. Unless you're, well, some plants use it. Okay. So now we have sex cells, we'll put them together. If you're making egg cells, when you do the divisions varies a lot from one living thing to another living thing. And in mammals, in our group, Meiosis I doesn't happen until the egg cell is released from the ovaries. While it's in the ovaries, it's pulling in lots of food, and it's kind of busy. It doesn't want to be doing something else complicated. It doesn't want to be doing a division, so it doesn't divide yet. When it's ovulated, it's got a little bit gap in time. It's not picking up food, it's not closing out extra sperm, and it'll do one division and form a polar body. Then it's in the oviducts where sperm are trying to get into it. And it's got to be able to let one sperm nucleus in and then completely block all the other ones. There's an enzyme interaction that it uses to block them. And it doesn't want to be divided while it's doing that either. So in mammals, what actually gets fertilized isn't technically an egg cell yet because it's got one set of double-stranded chromosomes, copies in its nucleus. So the sperm nucleus gets in, only the nucleus because the sperm has a mitochondrion. and mitochondria have their own DNA, and sometimes sperm mitochondrial DNA and egg cell mitochondrial DNA don't get along together if you combine them in the next generation. Sometimes they have conflicts. So it's kind of set up that the sperm mitochondria don't get in, just the nucleus. And then the sperm's like, okay, come on. It's the beginning of, male-female. And the egg cell's got to do its second meiotic division in order to free another polar body and wind up with a single strand, one set of chromosomes. Then the two get together, and that is fusion. The two nuclei, each with one set of chromosomes, fuse together, and what that cell is now is called a zygote. First cell of the next generation. It divides, and that forms an embryo. So the cells make copy of the cells, and they pull in food from whatever food supply has been given to them, and they make more cells and more cells and more cells and more cells, and eventually they wind up with a big ball of cells, and the ball of cells becomes hollow. I don't know if I've got this. Nope. So the ball of cells becomes hollow, and then the ball dents, and some of the outside flows to the inside and forms an inner layer paralleling the outer layer. If you're copying along with me, leave yourself some room. I'm not going to write it down here, but you're going to need to. So the ball puckers and you get a flow inward, and the cells that wind up staying on the outside is your ectoderm, outside skin. The ones that form the inside layer is your endoderm, your inside skin, and eventually you get cells in between the two. The middle layer, mesoderm, sort of means middle skin. And they're all going to form different parts of an embryo in most animals. The ectoderm is going to be what you would think it's going to be the skin, but it's also going to be the nervous system. Your nervous system starts as a plate down your back that eventually goes inside. So your skin and your hair and scales, if your feathers and that sort of stuff, are all ectoderm. If you're an insect, your skeleton, which is on the outside, comes from ectoderm. Your endoderm, your digestive system, right? It's your inner space. And stuff that grows off your digestive system, like breathing system, gills and lungs, and certain glands and other sorts of things that grow off of the digestive system. And mesoderm is a lot of other stuff, your muscles, your circulation system, your waste removal system. If you have a skeleton on the inside like we do, our skeleton comes from mesoderm. So a certain number of glands. Glands tend to be a combination of a bunch of things. But the different layers will eventually, it's a setup. Once you get the layers in place, you can start to have different cells use different genes and become different types of cells. They all have the same genes. They're not all going to be using the same genes. This is called differentiation. Some of the first genes that activate, other than the ones that are making cells divide, are called the Hox genes. Originally called the homeo genes, then the homeobox genes, then the Hox genes, all capitals, and now you tend to see it capital H, little o, little x. But they've been changing, messing with the name of it for 30 years. So if you read something old, you'll see different versions of this. Your Hox genes are your basic layout genes. They start to activate, and when they activate in one place, that's going to be the front end. Different genes activate, that's going to be the rear end. Different genes activate, that's going to be appendages. Or they're going to differentiate right side from left side. They're going to go, okay, this is where the nervous system is going to form. This is where the heart is going to form. So your Hox genes are your basic early layout genes that determine where stuff is going to be and how the embryo is going to be laid out as an eventual being. And then you get the development of tissues, you get the development of organs and organ systems and all that stuff, and you wind up eventually with offspring, right? We use the term offspring a lot. Okay, let me see. Yep, that's a good place to stop. There's a lot of stuff in this chapter. So, it's a lot of videos, some of which were on the last exam, a lot of which are going to be on the last exam. We're stopping here.